So we've talked a little bit about membrane structure already when we were talking about lipids, especially phospholipids. Hey, Nuri. So we know that the plasma membrane is a boundary that separates the living cell from its surroundings. So everything within the plasma membrane is the cytoplasm, which includes organelles. If you remove the organelles, what you have is the cytosol. And so the plasma membrane helps to make a cell separate from the rest of the environment. And the plasma membrane is selectively permeable. So some substances can pass through it, other substances cannot. Phospholipids are the primary type of lipid in the plasma membrane. And we talked about phospholipids having a polar head and a hydrophobic tail, so an oily, nonpolar tail. So that makes phospholipids amphipathic molecules. So they have both hydrophobic and hydrophilic regions, so both polar and nonpolar regions. The fluid mosaic model is the model that explains the structure of the plasma membrane. It is fluid, so there is movement within the membrane, and there's a mosaic of various proteins embedded in the membrane. So membranes are fluid. Right? Those phospholipids do move around, and there are many proteins associated with the membrane. Some are embedded in it and go all the way through from one side to the other. Other proteins are stuck on the top part of the membrane outside the cell. Others are stuck to the inner part inside the cell. So we've talked about the structure of a phospholipid. And remember, we've got that polar charged head, which is hydrophilic because it's charged. It can interact with the partial charges on the water molecules. Remember, water molecules have polar covalent bonds. And so the oxygen is a partial negative charge and the hydrogens have a partial positive charge. And so they can interact with the charges in the head. But the hydrocarbon tails are nonpolar. No charges associated with them. So they're hydrophobic. They don't like to interact with water. And the example is oil and water, right? Because oil is a lipid. It does not react with water. It tries to avoid water, minimizes its contact with water, which is why the oil floats on top. So when you mix phospholipids and water, they form this characteristic bilayer configuration where the hydrophobic tails face each other and the polar heads face the environments where the water is. So whenever you mix phospholipids with water, you always get a bilayer configuration because that's the most thermodynamically stable conformation. Phospholipids can move within the membrane. So again, it's a fluid, so things move, especially the phospholipids. Mostly they move laterally, so they stay in the same layer, right, that they're in. But occasionally they can flip and move from the outer layer to the inner phospholipid layer. So we call that a transverse flip. It doesn't happen very often because it's hard for that highly charged polar head to move through the interior of the membrane. But it does happen every once in a while. So they estimate about once per month there'll be a flip-flop, so a transverse flip where a phospholipid in one of the layers flips over to the opposite layer. But when it comes to phospholipids just moving around laterally within the same layer, that happens a lot. So they are in motion, right? Because your membranes are actually very fluid, a lot more fluid than you realize. And part of that fluidity has to do with some of the hydrocarbon tails having a kink in them, having that bend where we have a double bond. We talked about how that makes fats unsaturated if they have a double bond, but that helps to space the phospholipids apart, which keeps them more fluid. If there are no double bonds and there's no kinks or bends in the hydrocarbons, that allows for those membrane phospholipids to pack more tightly together, and that makes the membrane more viscous, much thicker. And so cells can regulate, this aspect of their cell membranes. And some of that has to do with temperature. Now, cholesterol, we know, is one of our lipids. It's one of the sterols or steroid lipids, and it is essential for the synthesis of vitamin D, as well as testosterone and one of the estrogens. It's also a lipid membrane component. So you do find cholesterol in lipid membranes, and it serves an important purpose there, as we'll see. So when it comes to temperature, cooler temperatures cause the membranes to switch from a fluid state to a more solid state. And so that's not ideal, right? Because we do want our membranes to stay fluid. So one of the ways to help deal with that will be to add cholesterol, or to add more kinks to the hydrocarbons in the phospholipid molecule. So membranes that are rich in the unsaturated fatty acids, right, the ones that have the kinks, that have the bends, right, where there's at least one double bond, those are more fluid. Whereas those membranes that are richer in the saturated phospholipids are more viscous. In general, your membranes do need to be fluid in order to work properly. And shockingly, they're about as fluid as salad oil. So that's pretty fluid, right? But we don't think of our membranes as being that fluid, but in fact, they are pretty fluid. And there are many proteins involved that we'll see that help provide integrity to those cell membranes and help keep cells stuck together. So cholesterol has different effects depending on the temperature. And so cholesterol is a way to moderate the fluidity of the membrane. So if temperatures are warm, like our body temperature is 37 degrees Celsius, then cholesterol helps to keep the phospholipids from moving around too much. So it makes the membrane a little less fluid because it's so warm. But when it comes to cold temperatures, and many animals live in cold temperatures, and animals that do not regulate their internal body temperature, like reptiles and amphibians and fish and things like that. So in a really cold temperature, the membranes would be way too viscous, way too thick. And so cholesterol helps to keep them more fluid because it prevents the phospholipids from packing tightly together. And so many animals can regulate how much cholesterol is embedded in their lipid membranes as a way to help regulate fluidity, either if it's too fluid or not fluid enough, right, by using cholesterol. So cholesterol is an essential nutrient, but it's something that we normally make as much as we need. So consuming excess cholesterol has negative health effects, as well as some people genetically produce way more cholesterol than they need, and that also can cause health problems. But cholesterol is essential. We need it. Right? We need it to synthesize vitamin D, right, the sex hormones, and we need it to maintain proper membrane fluidity. So cholesterol is good in the right amounts. Excessively high amounts, and it's bad. So proteins determine most of the membrane-specific functions. There are peripheral proteins and integral proteins. A peripheral protein is attached to the surface of the membrane. It could be the outer surface or the inner surface. Integral membrane proteins they go all through the membrane, so they penetrate the entire membrane. Integral proteins that span the membranes are called transmembrane proteins. So here we see examples, our phospholipid bilayer. Here we have some transmembrane proteins, maybe some that are just stuck to the top or the bottom. Now when it comes to a transmembrane protein, remember that the interior of the phospholipid bilayer is hydrophobic. It's nonpolar. And then the exterior has the polar heads and the watery environment. So a transmembrane protein is both polar and nonpolar. It's also one of our amphipathic molecules. The interior of the protein is nonpolar, so it's hydrophobic, so it can be in the middle of the membrane. But the exterior parts of the proteins that stick out into the watery environment, they're polar, so they can interact with the water. So a transmembrane protein is an amphipathic molecule. It has both polar and nonpolar regions to it. So the 6 major functions of membrane proteins are transport. As we're going to see, cells have to transport materials into and out of the cell, and proteins assist with that. There are enzymes that may be on the outside of the cells, attached to the membrane. Signal transduction, we've talked about signal molecules and their protein receptors. Cell-to-cell recognition. So cells in a tissue need to recognize the other cells in that same tissue. So your muscle cells know that they're in contact with other muscle cells. Your liver cells know they're in contact with other liver cells. So they do talk to each other, and there are connections between the cells which allow them to communicate, often by sending chemical signals, from one cell to the next. Intercellular joining. So cells, or the cells, need to stick together to form tissues and organs. And then attachment to the cytoskeleton and the extracellular matrix. So we talked about inside of a cell, a very complex network of fibers. Just on top of the cell is also a network of fibers called the extracellular matrix. Now back to the intercellular joining. Inter means what? What does inter mean?
Like within?
No. That would be intra. Intra is within. Inter is between. So it's on the outside. So it's between cells. So intercellular joining, there are several types of protein connections that happen. We have gap junctions, adherence junctions, tight junctions, and desmosomes. So these protein connections are why when you gently tug or pull at your skin, it doesn't rip apart, right? It stays together because of all these protein connections between cells. Because our cells are just membrane. Membranes are not rigid, right? So we need to have protein connections to hook them all together. And when you hook a bunch of cells together, there is strength in numbers. Of course, if you pull hard enough, you will rip those cells apart, right? You'll separate them, and that's how we get injuries. So, but these are the 6 major functions of membrane proteins. So here we see transport proteins. We're going to talk a lot about these in a couple minutes. We can have passive transport, where molecules simply diffuse through a protein channel. We can have active transport, which requires energy, like ATP. We have enzymes, which can break down molecules into smaller pieces or perform different functions, which could be on the inside or the outside of the cell. We've talked about signaling molecules and receptors right, which are made of protein. Cell-to-cell recognition. So in order for cells to know they all belong together in that tissue, in that organ, they do connect with different proteins so they can recognize each other. In some cases, like with the immune system, immune cells need to know that they are on the same team, right? They're in the same army so they can fight against a common enemy. And so there are cell-to-cell recognition molecules that your immune system uses in order to know they're all on the same team. Intercellular joining to keep those cells together to form that tissue or that organ, and then attachment to either the cytoskeleton inside the cell or the extracellular matrix. So many, many different proteins are associated with membranes. A cell must exchange materials with its surrounding, a process that is controlled by the plasma membrane. So we've already said that plasma membranes are selectively permeable. Some things can diffuse through the membrane, other things cannot. They need help. Hydrophobic, nonpolar molecules like hydrocarbons, they can typically dissolve through the lipid bilayer and pass through the membrane to get into and out of the cell. Also, things like oxygen and carbon dioxide can diffuse through the membrane. But if you have a molecule that's really polar, like sugars, water, those cannot cross the membrane easily because of that hydrophobic interior. And so they have great difficulty simply in dissolving through the membrane. So they need help. So transport proteins help molecules to be transported to the inside or the outside of cells. Some transport proteins are channel proteins, which have hydrophobic, parts where they are embedded in the membrane. So remember we said that some of the transmembrane proteins, that they're amphipathic, they got polar heads and a hydrophobic interior to associate with the hydrocarbon tails of the phospholipids. And so this can help polar molecules or ions to move through the membrane by using that protein channel. So we can think of it as a tunnel, right, which allows molecules to move through. When it comes to water, there are special channel proteins called aquaporins, which allow water to move through the membrane easily, right? Because they're charged. Water molecules are polar, so they can't get through that hydrophobic interior, but there are special protein channels called aquaporins, which allow water to diffuse through. There are other transport proteins called carrier proteins, which do up to transport molecules across the membrane, but no energy is required. It's just that particular channel protein can interact with a molecule, causing the channel protein to change its, or the carrier protein to change its shape and allow it to move that molecule through the membrane. But no energy is required to do this. And those particular transport proteins are specific for whatever is being transported. So molecules are always in motion, right? So molecules are always moving. When it comes to cells, we are talking about an aqueous environment. So aqueous means watery. There's water inside the cell, there's water outside the cell. Everything is moving in the water. When it comes to diffusion, that is the tendency for molecules to spread out evenly into the available space. So molecules diffuse from an area of high concentration to low concentration so that the concentration is equal everywhere within that particular space. So the molecules are diffusing to reach equilibrium, equal concentrations everywhere within that available space. And this is a natural property. It doesn't require any additional energy besides what those molecules have, right? Because they're vibrating anyway, they're moving around. And so they will diffuse from an area of high concentration to low concentration. An example you might be more familiar with, if there's someone walks into a room and he or she is wearing a strong perfume or cologne, after a certain period of time, everybody in that room will be able to smell it because the molecules are diffusing from an area of high concentration, which is that person that has the perfume or cologne, and spreading out through the room, right through the air. It's diffusing, and eventually everyone will be able to smell that perfume or that cologne. So that's an example of diffusion, through the air. So molecules will diffuse through the air from high concentration to low concentration. In an aqueous environment, molecules diffuse in that watery environment from an area of high concentration to low concentration. And the goal is to reach an even concentration everywhere in that available space. At that point, equilibrium has been reached. And so no more diffusion will occur, right? Because the concentration is the same everywhere. So when we have a membrane, and if it was a cell, it would be the lipid membrane. In the lab, we use artificial membranes to demonstrate this process. And so water can easily pass through these membranes in lab. And so if you have a solute, in this case, a dye molecule, so a colorful molecule, if you put a drop of food coloring into a beaker of water, it's very, very dark where the drop initially was in the water, but then eventually the dye molecules will spread out evenly all throughout that beaker, and you'll have an even concentration. And that will happen naturally. Of course, you can speed it up by stirring up the water, but if you just leave it alone, eventually those dye molecules will spread evenly throughout that beaker of water. And so we're seeing that here because the dye molecule is small enough to get through the membrane. It will move across to the other side and reach equilibrium, reach an even concentration. Now, the dye molecules are still moving, and some will move to the right, some will move back to the left, but there'll be no net movement, no net change, right? The overall concentration is now equal on both sides of the membrane, and it'll stay equal. If you add more than one solute, if they can get through the membrane, they will move right to the other side until an equilibrium is reached. We have equal concentrations of both solutes on both sides of the membrane. So as long as a solute molecule can diffuse through the membrane, it's going to try and reach equilibrium on both sides of the membrane. But not everything can get through membranes, whether it's this artificial membrane in lab or actual cell membranes. And so that causes some interesting things to happen. So we already mentioned that molecules diffuse from an area of high concentration to low, so they diffuse down their concentration gradient. And no work is required to do this. No energy other than that which exists in that system is used. And once molecules have reached equilibrium, there's no net diffusion, there's no net change. So we call this movement of substances across the membrane, right, by diffusion, passive transport. And so molecules can diffuse into and out of cells as a result of following their concentration gradient from high to low. They may not be able to diffuse directly through the membrane without help, like a protein channel or a carrier protein. But because no additional energy, no ATP is being used is considered passive. Active transport requires ATP, as we'll see shortly. So all molecules will diffuse following their concentration gradient. That includes water. Water can move from an area of high water concentration to an area of low water concentration in an attempt to reach equilibrium. We call the diffusion of water across a selectively permeable membrane osmosis. So osmosis is a special case of diffusion involving water and a selectively permeable membrane. So here we have the original YouTube, not what you watch on your computers and smartphones, but a glass tube that is bent in the shape of a U, and there's a selectively permeable membrane at the bottom in the bend. And so water can diffuse across this membrane, but the solute, like a sugar, which is a big molecule, cannot diffuse across the membrane. So if you put one solution on one side that has a low concentration, so let's say it's a 1% sugar solution, on the other side you put a 10% sugar solution, so you got a lot more sugar dissolved in the water. On the right side, it's a higher concentration of sugar. The sugar would like to go to the other side and make the concentrations of sugar equal on both sides of the membrane, but sugar cannot diffuse through this membrane. The sugar is too big. It can't get through. The only thing that can get through is the water. And so water moves into the high solute concentration. And that's something to memorize. Water always moves into the high solute concentration. So the solute is whatever is dissolved in the solvent, which for our purposes is always going to be water. So the high solute concentration is where the water moves into. And so water will move across the membrane in an attempt to equalize the concentration of sugar. Right, so concentration, it's the amount of solute divided by the water. Right, so it's the amount of sugar divided by the water, and that gives you the concentration. So because water is the only thing that can move, it moves into the high sugar concentration, into the high solute concentration, and the water actually defies gravity and moves up the tube. So by adding more water to this side, we are diluting that sugar solution, making it less concentrated. And so ultimately, in using my examples here of a 1% solution on the left and a 10% solution on the right, enough water will move across to the right side to reduce the concentration of sugar from 10% to 1%. And that way the concentrations will be equal on both sides of that membrane. But the water moves to achieve that. So water always moves into the high solute concentration. Another way to remember that, which I don't think is as easy, but water is following its concentration gradient. If we have a 1% sugar solution on the left, that means there's, 1% of sugar and 99% water. On the right, if we have a 10% sugar solution, it's 10% sugar and 90% water. So 99% water on the left, 90% on the right. There's a higher concentration of water on the left. It moves, it diffuses into the area of low water concentration in order to make the concentration of water equal on both sides of the membrane. So either way that you remember it works. I just think it's easier to think of water always moving into the high solute concentration. Or you can look and say, okay, where's the concentration of water greater? And the water is going to move to an area of low water concentration. So either logic works. I just like to remember water moves into the high solute concentration because it's easy to remember that. So any questions on where the water is going to move? No, all good? No. So we're trying to achieve equal concentrations on both sides of the membrane. Whether you choose to think about equal concentrations of water, which is a solvent, or equal concentrations of the solute, whatever that is that's dissolved in the water. Either logic works. Like I said originally, if we have a 1% sugar solution on the left, and then we have a 10% on the right, we have a higher concentration of sugar on the right. The sugar would want to move to the left, but it can't get through the membrane, right? Because the sugar wants to diffuse from an area of high concentration to low, right? From 10% to 1%. But the sugar cannot get across the membrane. It's stuck. The one thing that can move is the water. And so water always moves into the high solute concentration. So it will move from left to right. And so the water molecules are diffusing to try and reach an equilibrium concentration, the same concentration on both sides of the membrane. And so the water will diffuse until it achieves equilibrium. And that means the water will move up our tube because water is moving into the high solute concentration. And it will do this on its own. No energy is required to make water defy gravity. It's trying to reach an equilibrium concentration. And the only way that it can do that is by diluting the sugar solution on the right. And so water will continue to move to the right until the concentration of sugar is the same on both sides of the membrane. Now, you can think of it in terms of water following its own concentration gradient, because if there's 1% sugar on the left, that's 99% water. That's percent. And then if it's 10% sugar, it's 90% water. And so water will diffuse from high concentration to low. So water is going to move from left to right. It is, oh sure, if your tube is not big enough or long enough, right, the water will start to spill over. So it depends on what concentrations of your sugar solutions you use, but the water will continue to move, right, to try and reach that equilibrium. So yes, the water can overflow. And we used to do this as an actual demonstration in lab. And sometimes as it sat there throughout the whole lab class, it would kind of overflow because it would, move too far up the tube. But we would have it moving up a tube, maybe 2 feet, 1.5 meters up the tube. So water can move up a lot. And we're talking about a relatively skinny tube, not a big fat tube. But the water will move. When you put, most guys will be able to relate to this, but if you've ever had a slug, that slimy, insect, well, it's not an insect, but it's a mollusk, but a slimy creature that's outside crawling around the, your porch or sidewalk or something, and you put salt on it, and the slug melts. It's because the water is being sucked out of the slug, right? Because those salt crystals, once you put them on that slug, is a very, very high concentration of solutes. And so water moves out of the slug into that salt to help dilute it, and it melts the slug and kills it. If you put salt on a wound, yes, it hurts. It also gets wet because the salt is sucking water out of your tissue. So water moves into the high solute concentration. Is that better now? So tonicity is the ability of a solution to cause a cell to gain or lose water. So cells are in a watery environment, right? They're in an aqueous environment. So the concentration of solutes has a big influence on those cells. An isotonic solution is one where the solute concentration inside the cell is the same as the outside of the cell. As a result, there's no net movement of water. Some water will move into the cell, some water will move out of the cell, but it's the same amount, so no net movement. And for many cells, especially animal cells, that's a happy condition. Eating too much salt can draw water into your blood vessels to increase the pressure, yes. Although your kidneys usually do a good job of processing that so you can excrete it, but we do tend to absorb the salt because salt is important. We need salt. It's just that too much salt is bad. Right? So all these things that are good for us are only good in the right amount. Right? Too much is usually bad. A hypertonic solution is one where the solute concentration outside of the cell is greater than inside the cell. Water moves into the high solute concentration, which means water will leave the cell and cause it to shrink. A hypotonic solution is one where the solution outside the cell has a much lower concentration of solute relative to the inside of the cell. And so water moves into the high solute concentration, which is on the inside of the cell, and so water moves into the cell, and that can cause the cell to explode, especially if it's an animal cell. So in an animal cell, if you put red blood cells into pure water, they explode because the water is moving into the cells, because the cells have a higher concentration of solutes relative to the pure water. If you put those red blood cells in the isotonic solution, they're happy. That's ideal. No net movement of water. And if you put cells in a hypertonic solution where the concentration of solutes outside the cell is higher, water leaves the cell, and so it shrinks and shrivels. So obviously that's a bad situation if it's red blood cells. So then when they give you an IV, it's not pure water. If they injected pure water into your blood vessels, your red blood cells would explode and you would die. And if it's a little bit too salty, your red blood cells would shrivel and shrink, and they would clog your capillaries and lead to organ failure, and again, you would die. So an IV must be isotonic. And so an IV of isotonic saline is salt water. Sodium chloride is technically 0.85% sodium chloride. So it's less than 1%, and that's isotonic, and that's ideal. Any more than that is going to cause your cells to shrivel, If it's significantly less than 0.85%, your cells will swell and burst, right? They'll explode. So IVs have to match the concentration of solutes in your bloodstream. Otherwise, the IV will kill you. Now, when it comes to animal cell or plant cells, the situation is a little bit different. Plant cells actually like a hypotonic environment. When you water your plants, you water them with, pure water, right? You don't water them with salt water because that will kill them. And so we need the pure water. It moves into that plant cell, which causes that cell to expand and push against the cell wall. And that makes those cells turgid or rigid. And that's why plants can stand upright, right? Because water is moved into their cells and causes them to swell up and create internal pressure. Now, the cell wall keeps them from exploding. Otherwise, they would explode. But the cell wall helps to prevent that. But it makes the plant cells rigid so the plants can stand up. If you use an isotonic solution, which means there's a little bit of salt in it, then the water is not going to rush into the plant cell wall. It's not going to make it nice and rigid or turgid. So it's going to get soft. It's going to get flaccid. And so the plant will start to wilt because it doesn't have enough, fresh water. If you use salt water or salt, it causes the plasma membrane to shrink and pull away from the cell wall. So the plant cells are not going to shrink that much because they have a cell wall, but the cell membrane pulls away from the cell wall, and we actually call that plasmolysis or plasmolysis. That's why you see plasmolyzed. And so plasmolysis will kill a plant. And so that's why in the old days, when you had invading armies and they would salt the earth, they put down a bunch of salt, and that meant that those people could not plant any crops because they would die from the salt until it rained enough to, dilute all the salt and it washed away, but that could take years. So this is what happens when you put different cells into these different kinds of solutions. What's perfect for a plant cell will kill an animal cell, which is fresh water. What is not great for the plant cell, which is perfect for an animal cell, is an isotonic solution. And then a hypertonic solution with a lot of solutes, usually salt, but also sugar, and that's bad for both plant and animal cells. So we know that there are organisms that live in fresh water, right? There are many protozoa, many single-celled little animals that live in lakes and ponds, right, which is fresh water, which means the concentration of solutes inside of those animals is higher than the external environment. And so water moves inside of these poor little single-celled animals, like paramecia. Well, paramecia do not have cell walls, so they need another adaptation to survive in a hypotonic environment where the water is constantly moving into that paramecium. And that's a special organelle called a contractile vacuole. And we're going to watch a video of this in a few seconds. But here you have a paramecium. Here is the contractile vacuole swollen with water. So it's sucking up the water that's moving into the cell, and then it contracts and squirts the water back outside. And here you see it contracted. It looks kind of star-shaped. And so that's the contractile vacuole in its contracted state. But it constantly swells and sucks up the excess water and contracts and squirts it back on the outside. So that's how paramecia can live in a freshwater environment. So let's look at a little video of this. So I gotta share my screen. and then go back. So here we're going to watch a paramecium and its contractile vacuole. So you'll see the vacuole expand and swell up, and then it will contract. See there? Here it is. It's now swelling up. It's filling with water, and then it's going to contract. Squeeze the water back out. And so the paramecium has to do this constantly. And there's more than one contractile vacuole in the paramecium, but it has to constantly pump out the excess water which is moving into it. Because the paramecium is a cell. It's got a lot of stuff inside. It's got a lot of solutes inside. And when it's in fresh water, water moves into the paramecium. So all swollen with water, and then it contracts and squeezes it out. So this is an adaptation which allows the paramecium to survive in a hypotonic environment. And we see the cilia beating over here, the cilia that it normally uses to swim, although it's being kept in place so we can watch the contractile vacuole contract. So this is an adaptation of a freshwater microorganism which lives in a hypotonic environment. So any questions on the paramecium and how it survives? Why the water moves inside?
Can you go back to the slide that was explaining hypertonic and hypotonic?
What, this one?
No, the one with the words. Yeah, this, okay. I was trying to figure out which one was greater. I know one is greater and one is less, but I'm trying to figure out, it says the concentration is greater.
Hyper means more, like hyperactive is more active, so hypertonic means more solute, and it's in the solution relative to the other side of a membrane. So it's always important to talk about where the hypertonic solution is, where the hypotonic solution is, because if you put cells in a beaker of 5% salt water, those cells are going to die because that's way too concentrated salt. So those cells are going to shrink and die because the water is going to move out of the cell into that hypertonic environment. But if you put those cells into pure water, then the concentration inside the cells is now hypertonic relative to the outside, where the outside is hypotonic relative to the inside of the cells. So it's all relative to the membrane. So you just need to know what side of the membrane are you talking about as to which one's hyper and hypotonic.
Thank you.
Okay. So we talked about the plant cell wall. Water moves into the plant cell. It causes the plasma membrane to push on that cell wall and make that cell turgid or rigid. And that's the normal state for a plant cell. So that's why rainwater or fresh water is used to water your plants, right? Because they need the water to move into the cells and create that internal pressure, right, to make them rigid. If they start to run out of water, right, then there won't be any water to move inside of the cells, and the cells get flaccid, they get softer, and the plant starts to wilt. If wilting goes on for too long, then the plant will die. So if it's just starting to wilt, you add more water and it perks back up. And this is something that you can often see in the summertime with certain plants that are outside, where in the heat of the day, when that full sun is just shining on them, and they start to wilt, because unless it's raining or it just rained or you just watered, they're not going to be able to get enough water into all those plant cells. And the plant cell wilts until the late afternoon or evening when the sun is no longer shining on them. And then you'll see the plants start to perk back up. So this is sort of natural. What happens with certain plants in the heat of the summer with full sunshine is they get a little bit wilty. And as soon as the sun is no longer directly overhead, the plant starts to perk back up because they're able to get enough water. But that's why if you have certain plants in your garden, if it doesn't rain enough, you have to water them because they will start to wilt first and then they will die. So again, we're now just looking at the plant cells. If you put a plant cell in a hypotonic environment, water moves into the cell. It causes the cell to expand, to put pressure on that cell wall, and that makes the plants turgid or rigid. And that's the ideal condition for plants, right? So they like hypotonic water. In an isotonic solution where the concentration of the water on the outside is the same as on the inside of the cell, there's no net movement of water. That's bad for plants. They start to wilt because they need water to move into the cell. If there's no net movement, then those cells will get soft, right? They'll get flaccid. And then if you use a hypertonic solution like salt water to water your plants, then the plasma membrane shrinks and pulls away from the cell wall. And we call that plasmolysis. So if you live near the beach, near the ocean, you are restricted in terms of the kind of plants you can grow there because, there's salt in the soil that is next to the ocean. And there are certain plants that have adapted, to tolerate, certain amounts of salt in the soil. But most everything else is not adapted to that. They can't handle that, so they'll die, which is why you're limited. If you have a beachfront property, you're limited in terms of what you can grow there, right? Because it's salty, it's sandy, it's not ideal, for most plants. But there are things, if you've ever been down to the Caribbean, you may have seen mangrove trees, or down in Florida, in southern Florida, mangrove trees grow in salt water, right? Because they have adapted to deal with that hypertonic environment. So there are different adaptations for plants and animals when it comes to dealing with salt. A saltwater crocodile, it excretes the excess salt, from little glands on top of its head, like its tear ducts, and it excretes the salt, excess salt that way because it lives in salt water. When it eats or drinks, it takes in salt water, so it has to excrete that excess salt. And if you're ever stranded at sea, you can't drink the ocean water because that's really bad. It makes your dehydration even worse. And so you'll die a lot faster if you drink the salt water, if you don't get any source of fresh water to drink. And human beings, they need a certain amount of water every single day to survive. You can live a lot longer without food, but you can only go about 3 days without water. And then you die because not enough water for your cells to function correctly. So we need water, but can you drink too much water?
In a short period, yeah.
Yes, you can.
Oh yeah. You can drink yourself to death if you drink too much water in a short period of time, right? Because that dilutes, your ions in your bloodstream and it upsets the balance. And so, yes, water intoxication is what they call that. So normally you think of intoxication associated with alcohol, but water is intoxicating, but not in the same way. So water intoxication will lead to death. So you can drink too much water in a very short period of time, enough to kill you. So we need a certain amount of water, especially if it's hot, if you're sweating, if you're physically active, you need more water. But we need a certain amount of water every day. But too much is actually a bad thing. Not enough is bad. So again, water is a good thing, but too much of it is bad.
I have a question. If you drink a lot of water in a short period of time, why can't you just, filter it? The kidneys don't filter it.
Well, they can only filter at just so fast a rate. And if you're drinking more water than can be filtered by the kidneys, then it disrupts the balance of ions in your bloodstream and in your cells. And that screws up all kinds of biochemical reactions, and so you will die. Your kidneys can only handle just so much water. And so too much water, and they can't handle it. And so you die from water intoxication. But that usually takes a lot of water. Most people don't drink anywhere near that much. And many people tend not to drink enough water. They always say thirst is not really a reliable sign of when you need to drink more water, because sometimes you don't feel thirsty even though you're becoming dehydrated, and by the time you feel thirsty, that means you're significantly dehydrated. So headaches can occur if you're dehydrated, and so if you suffer from headaches, not migraines, but just ordinary headaches, try drinking some water and see if that doesn't help. And just something that people don't think about in the wintertime, because in the wintertime it's cold and you're not sweating, but you are dehydrating because the air is dry and you lose water, through exhaling, water through your skin. And so you can become dehydrated in a cold, dry environment without realizing it, because most people think, well, I normally drink a lot more when I'm hot and I'm sweating, it's cold, I'm cold, and yet you can still become dehydrated because you don't feel that thirst signal until you're really dehydrated. So don't rely on thirst as being a reliable indicator that you need to drink more fluid. So now we're going to talk about how cells can move things back and forth through the membrane. And so we have a facilitated diffusion in terms of passive diffusion, right? Those channel proteins or the carrier proteins. So this is passive. No extra energy is required. With a channel protein, molecules in a high concentration will diffuse across the membrane to an area of low concentration, or just following your concentration gradient. A carrier protein also helps that to occur. No extra energy is required for a carrier protein to do this, though. It's all passive. Also known as facilitated diffusion. But sometimes cells want to concentrate molecules against their gradients. So they want to pump molecules from an area of low concentration into an area of high concentration. That's not the way molecules work, right? They diffuse from high to low, but the cell wants to concentrate that substance from low to high. That requires the input of energy, and that energy is in the form of ATP, usually, and so this is an active process. So active transport will move substances against their gradient from low concentration to high concentration. That's not the normal way things work. So cells have to input energy in order to make that happen. And that energy is typically ATP, but not always. Active transport is performed by very specific proteins embedded in the membrane. So here we see passive transport. Certain molecules, nonpolar molecules, small things like oxygen and CO2 can diffuse through the membrane. Other molecules that are either big or maybe charged, they need help. A channel protein, which allows molecules to diffuse from high concentration to low, or a carrier protein, which does the same thing, except that the molecule that is the carrier protein is designed to pick up and transport a substance from one side of the membrane to the other, but without using any extra energy for that. It's just the nature of that molecule. And then active transport requires energy, usually ATP, because cells are trying to concentrate a substance against its gradient. Now, remember the active transport, because when we start getting into aerobic cell respiration and learning how cells make ATP, in order for cells to make a lot of ATP, there needs to be an alternate source of energy for that. Because if you're trying to make a lot of ATP, you cannot use ATP to make ATP, right? There's no net gain. And so there needs to be alternate forms of energy. As we're going to see, one of those alternate forms of energy is the stored energy in electrons, which will be used to pump hydrogen ions against their concentration gradient. And then we're going to see that that high concentration of hydrogen ions is itself an alternate form of energy that allows an enzyme to make ATP. So it will be a remarkable system that we're going to study, where alternate forms of energy are needed by the cell in order to make ATP. And we're going to see molecules being pumped against their concentration gradient. And then we're going to see molecules following their concentration gradient in order to make ATP, but we'll save that for later. So small molecules will diffuse through the membrane, or a channel protein, or a carrier protein, or perhaps active transport. But sometimes the cells want to move a large quantity into and out of the cell all at once. We call that bulk transport, and this also requires energy for that to happen. So exocytosis is the process of leaving the cell, and so a transport vesicle that may have originally come from the endoplasmic reticulum, and then it moved to the Golgi, and it budded from the Golgi, and it's now heading for the plasma membrane. That vesicle is carrying substances to be secreted by the cell. And so the vesicle fuses with the plasma membrane so it can release the contents to the external environment, right? But transporting something in a vesicle requires energy. Endocytosis is the opposite. You're moving from the outside to the inside of the cell. And so there are three types of endocytosis. Phagocytosis, which I mentioned before, cellular eating, where a phagocyte, a cell, engulfs something, often to destroy it. Penocytosis, cellular drinking, so sometimes cells need to move more water into the cell than can occur with just the aquaporins. And then a receptor-mediated endocytosis. And so this is a very common way, where cells have receptors on the outside of the plasma membrane. And when a molecule called a ligand binds to that receptor, it causes the cell to engulf and to move in a large quantity of ligands, right? Those molecules that the cell wants to bring to the inside. Unfortunately, many pathogens, many disease-causing organisms, like bacteria and animal viruses, exploit this natural process in order to infect cells. And I did mention this last time as well. So a virus lands on one of these receptors, and the cell just thinks, oh, goody, this is something that I want to eat. And so the cell engulfs the virus and moves the virus from the outside to the inside, which infects the cell. And so cells actually infect themselves with the virus. Because viruses are exploiting this natural process. They're tricking, right, that cell into infecting itself. And we're going to watch a little short video here. I just want to get through these last slides. So here we see receptor-mediated endocytosis. So whatever the substance is the cell thinks is good to eat, right, the ligand, it binds to the receptors, and that causes the membrane to pinch inward and form a vesicle. So it's just moved a large quantity from the outside to the inside of the cell. And this is receptor-mediated endocytosis. In the case of viruses, the virus has landed on this receptor, and so the cell has engulfed that virus. It's moved that virus from the outside to the inside. Now this cell has been infected. Okay, so let's go watch that video. And it's one that I've been showing for years. It's especially relevant now because of COVID-19. It shows the infection of a cell by a virus. It's only about 3.5 minutes long. So.
A virus is a small infectious particle that hijacks the machinery and metabolism of our cells to grow and reproduce. Each viral particle, or virion, consists of nucleic acid, either DNA or RNA, in single or double-stranded form, enclosed by a capsid. In most viruses, the nucleocapsid is enclosed by an outer envelope. Influenza type A, an enveloped single-stranded RNA virus, has been widely studied and serves here to illustrate the life cycle of one common virus. Scientists believe that influenza virus is spread from person to person through exposure to large respiratory droplets, direct contact, or airborne dispersal. Infection takes place mainly in the respiratory tract. Infection begins with attachment of virus proteins to a receptor on the surface of the host cell. The virus is then taken in the cell by receptor-mediated endocytosis. As the virus enters the cell, the virus is internalized in a membrane-bound capture vesicle that carries the viral core. The vesicle is transported on microtubules inside the cell by host proteins called kinesins. During transport, the membrane of the vesicle fuses with the membrane of the virus, and the capsid undergoes uncoating. The viral core RNA and proteins are then released into the cytoplasm, where they are guided by host proteins to the nucleus of the host cell. At the nuclear membrane, the viral core uses host protein channels to enter. Inside the nucleus, cell machinery is utilized by the virus to replicate the viral genome and make messenger RNA, mRNA. Some viral mRNA exits the nucleus to exploit cellular ribosomes to direct synthesis of viral proteins. Viral proteins go back to the nucleus to associate with viral RNA. These nucleoproteins again leave the nucleus and use cellular processes to travel to the cell surface. Viral surface proteins are made and processed in the cytoplasm and also travel to the cell surface, where they combine with encapsulated nucleoproteins to form progeny viruses, which depart from the cell by budding. The virion now goes on to infect other cells. At present, vaccines to stimulate the production of antibodies against the virus and stimulate host-cellular responses to ingest the virus remain the best strategy for controlling certain viral diseases. but they are of limited effectiveness against rapidly mutating viruses and are ineffective against viruses that cause the common cold and AIDS.
So budding
Viruses to ingest the virus remain the best strategy for controlling to infect other cells.
is the common way of
Encapsulated
saying
nucleoproteins to form
exocytosis.
progeny viruses which depart from the cell by budding. The virion now goes on to infect other cells
And then you'll see a white blood cell
and
reaches
stimulate
out
host cellular
and
responses
grabs
to
the
ingest
virus
the
to
virus
destroy it.
remain the best strategy for controlling certain viral diseases. But
Right, so there just reached out a white blood cell to grab that virus, to engulf it and destroy it, right, by phagocytosis. So we learned about endocytosis, right, for the virus to get in, exocytosis or budding for that virus to leave. There was also, let's see.
Go back to the nucleus to associate with viral RNA. These nucleoproteins again leave the nucleus and use cellular processes to travel to the cell surface.
So endoplasmic reticulum, Golgi, right? So as the viral proteins are processed, so viruses, they take control of a host cell and they force that host cell to replicate new viruses. But we were able to see endocytosis, exocytosis, the Golgi and the ER, budding, which is a simpler word for exocytosis. So you can relate that video to COVID-19 because it's the exact same idea, right? Where the host cell engulfs the COVID-19 and infects itself. It's a respiratory infection, although it can cause a lot of other problems elsewhere in the body, not just the respiratory tract. So, but any questions about the cell membrane, osmosis diffusion, not on the exam tomorrow. So any questions about the exam tomorrow? Starts at 2 o'clock, Montgomery College time. So if it's 2 o'clock in your home and you don't see it yet, just keep refreshing and it will appear. And you have the whole class period to take it, which is extra time compared to the normal class. Yeah, there's, I think, 1 or 2 short answer questions. And that's, oh, spring break is done. We're past spring break. Our class started after spring break, so spring break is over. Right, because we don't follow Easter spring break like the public schools. Our spring break is in mid-March, which is true of most colleges and universities. So you get no breaks in this class. It's 7 weeks straight from start to finish. Around 50 questions. If it were all multiple choice, it would be 50, but there are some written questions. Yes, you must be logged into the class. As I said, I'll have an open room available, so if you have a question, you can join the room. You have to ask me the question, otherwise you'll just be taking it. And you have the whole class period, which is more than what we normally get. Double check the syllabus or course content. It gives you all the chapter numbers. The exam will appear under the course content. And let's go back and look and see. So if you go to course content, and it's the second week, number 2, it says exam. 1 and then metabolism, energy, and enzymes, which will be the lecture we do on Thursday. But when you click on that course content, it will take you to the exam. But as I said, it won't appear until 2 o'clock McCormick College time. No, do not turn on your cameras or microphones. You just take it. I will be there. If you need to ask me a question, then you can join the room and turn on your microphone to ask me a question. So this is where I put my trust and faith in you guys, right? Because the first exam is normally where I catch the first cheater. So, right, you only need to join the room if you have a question. Absolutely no Google, no textbook, no notes. It's just like you're sitting in the classroom, except you're sitting at home, but you're expected to take your exam just like you would normally take it, sitting in a classroom. I know temptation is great, but well, I see this, Lauren, so any questions you've asked, hopefully I've seen them. So avoid the temptation. I know it's very easy, and it's so easy that you do something you shouldn't do, and then you get caught, and then it's all over, right? So avoid temptation. Hopefully you've been studying all along, right? So you have your little group me where you can get together to ask questions of each other. Yep, under the week 2 course content, but the second week 2 module. It says exam on it, so you'll see that. And you have plenty of time, so if you don't start till 2:05, you're not late, right? Because you have extra time. Is lecture over now? Is that what you're asking, Crystal? It's over except for questions. Yes, you must log into the class, otherwise you can't access the exam. So you got to log into Blackboard, under this class to get to the course content. You just don't have to log in to collaborate, right? Unless you have a question. Right, so I'll be there in that open room. So if it's 2 o'clock and you have a question, before you start the exam, you can ask me then. So, but like I said, you guys have extra time because it's the same exam. It's just that in the 7 week, we have a longer lecture period, so you get extra time. But yes, the exam will become available at 2. No, there should be a little timer there when you start the exam, so you know how much time has elapsed. But if you want to have a clock, next to you, or a watch, that's okay. It is a good idea to submit after you answer each question, just in case you get disconnected. Everything will have been saved. If for some reason you do get disconnected, when you reconnect, either join the room so you can talk to me or send me an email. Our lab's going to start today at 3:30 as usual. Yeah, lab should not take the whole period today. It's just a microscope. A few calculations, nothing major. All right, so I'll see you guys at 3:30.
Thank you.